Okay, so today is Wednesday, Friday, what's happening? That's on Friday, so we're gonna spend some time, we're gonna finish talking about the fourth macromolecule today, and then we're gonna talk some about the test. Reminder, also on Friday is department picnic. If you're a member of our department, you should have gotten an email from Samantha Herbert inviting you to come. Please RSVP so we can make sure you have a. Also thinking about next week, I am muted.
I think I know that. I think I'm okay with that.
Next week, your chapter 6 homework is originally gonna be due on Monday. I decided to send a test on Friday that we would just make a homework due on Wednesday. So that has not yet been changed. I'm gonna try to change that today, but just letting you know, the chapter 6 homework, which I'm hoping to post this afternoon or tomorrow morning, will not be due until Wednesday, the 18th. Okay? Questions about any of that? Okay.
Oh, that's a reminder, the department picnic. Sam made a pretty slide for us. Okay, I forgot to bring something.
Okay, this is where we left off yesterday, Wednesday, Monday, whatever day it is, I don't even know. Don't see if anybody got any of these. So you had that handout that had all the 20 amino acids on it. You were looking at those 20 amino acids. You were looking at how many carbons they had, finding the amino group, what's the other group that you put on there? Box. All amino acids have that alpha central carbon. On the left-hand side of all of these, at least, you had the amino group, and on the right-hand side is what? Carboxylic acid. So that's C double bonded to an O and an OH. And then you have an R chain that makes it unique. So you are looking at those R chains then and determining characteristics of them. Are they hydrophobic? Are they hydrophilic? Are they acidic? Are they basic? And remembering that that's going to affect the way they interact in which level of protein structure. Which level of protein structure does the amino acid side chains, the side chains of the amino acid, their interactions, which level does that determine? Secondary is going to be hydrogen bonds forming between the backbone, that amino carboxylic acid backbone. So that's going to be your alpha helixes and your beta pleated sheaths. Tertiary is where we're going to see the interactions between those different side chains. And we talked about those on Monday. Those different, the ones that are hydrophobic are going to tend to do what? Where are they going to be on the protein?
Inside.
Yeah, they're gonna want to be inside, away from the water, because they're in the cell. Cells are mostly water, so they're gonna want to be away from that water, so they're gonna tend to fold inside. When they get next to each other, if they're really close to each other, because those electrons are moving around everywhere, and they may not be evenly distributed in the orbitals, what kind of interactions can we get? If the electrons are not evenly distributed in the orbitals, you might have a little bit of positive over here and a little bit of negative over there. What kind of interactions, when they're really close together, does that cause? Not strong enough to be a bond. The van der Waals interactions coming from those temporary positives and the temporary negatives that are coming from the movement of the electrons around there. So hydrophobic amino acids that are like close to each other. Hydrophobic amino acids that are close to each other as they fold on the inside of the protein might have van der Waals interactions between them. So that's one way they can interact. How else can side chains interact? We talked about 4 ways. Van der Waals interactions. Can't look back in your notes, okay, this is easy. That man. Besides, so the side chains, they can interact in 4 different ways. So one of them is the van der Waals interactions between hydrophobic ones. There's 3 other ways that side chains can interact in the tertiary structure of a protein. You can form a covalent bond between two sulfide, the sulfhydryl side chains can form a covalent bond. Use that sulfide bridge, the disulfide bridge. So that's a second way is the disulfide bridge.
You create a bond.
Hydrogen bonds, always hydrogen bonds, right? I mean, I feel like we talk about hydrogen bonds literally all the time. Because they do so much, they're so important in biological molecules, so. Covalent bonds, hydrogen bonds, van der Waals interactions.
So
And the salt bridge, which is an ionic bond that can form between an acidic and a basic. Remember, some of them are acidic and basic, that can cause the positive and negative charges, they can form an ionic bond through the salt bridge. Okay, and then there were some jokes here for you to think about. Just making you think about the names of the amino acids in a different way. So what did the amino acid say when he was asked the time? Did anybody get it? Yes, it's 309. You pronounce it 309, but it looks like 309. Oh, yeah. I told you they were bad jokes. What is a pirate's favorite amino acid? Arginine. Yeah. Where do amino acids go to pray? This is my favorite.
16.
The Sistine Chapel. That's my favorite amino acid. What amino acid does Ricky Ricardo love? I didn't know if this is like, who is Ricky Ricardo exactly? I didn't know. Does anyone know who Ricky Ricardo is? Anyone? What?
It's the experience.
No, I mean, maybe there, but I don't know.
That's.
He was married to Lucille Ball, so what's his favorite amino acid? Leucine. This is the favorite amino acid. I love Lucy. Yeah. That was a stretch, I know. What is the devil's favorite amino acid? The devil's, it is lysine because he sputters lies. Lysine. I didn't even make these up, just for. What is the hottest amino acid? Serine. Did you sear something? It's really hot. What is a snake's favorite amino acid? Okay.
Make sure at least think about the names of them. Okay. So we spent a lot of time on Monday talking about protein structure and the 4 levels of protein structure. And we talked about at the end, we were talking about the fact that if you change one of the amino acids in the primary structure, then you change the secondary and tertiary and sometimes quaternary structure, and that has major effects. So we're going to start talking about the 4th macromolecule by thinking about the fact that somehow we have to figure out what the amino acid structure is, what the amino acid sequence is. So what molecule determines the amino acid sequence? DNA. So when we talk about proteins and protein structure and amino acid sequence, we can't talk about it at least without a little bit of talking about our 4th macromolecule, which are the nucleic acids. So we're going to talk about the structure of nucleic acids, but before we do that, we're going to talk about the idea of the central dogma of biology. This is what we're going to cover in our last chapter of this semester, but we can't talk about it too much. We never talk about the central dogma of biology too much. Maybe you feel like we can, but I don't feel like we can. So DNA, you probably have heard of, you probably are a little bit familiar with the structure of DNA. You've probably seen the structure of DNA before, but DNA is literally the instructions, the code for how to build a protein. And how does it do that? DNA is found in the nucleus. And I think one of the questions on your homework asked you to think about differences between DNA and RNA. And some of you said DNA has the ability to self-replicate. So the process of taking DNA and making more DNA out of it is called replication. So DNA can make DNA. DNA can also make another nucleic acid, RNA. So we use DNA to make RNA, and then RNA is then translated into proteins. And by proteins, we mean the amino acid sequence, not because the DNA is just determining the sequence of the amino acids, and then those chemical interactions will determine the secondary and tertiary and quaternary structure. So the primary structure of a protein, its amino acid sequence, is determined by the nucleic acids in the cell, by the DNA, which is making the RNA, which is then making the protein. And that's the central dogma of biology. The central dogma of biology says DNA makes RNA, RNA makes proteins. And again, we're not going into detail about this now. We will go into detail about this, lots and lots of detail about it in chapter 11, I think it is. But I wanted to expose you to that idea of the central dogma of biology and thinking about the fact, again, it's all interrelated. We can't talk about proteins without talking about DNA, and we can't talk about DNA without talking about proteins because they are closely related to each other and they help determine each other. So when we talk about nucleic acids, again, this is our fourth macromolecule. So we had carbohydrates, lipids, proteins, and nucleic acids is our last one. It's a polymer, so it's a true polymer. So what are polymers made up of? In general, you make them out of monomers. So when we talk about the monomers of DNA and RNA, the monomers are called nucleotides. So this is showing you one monomer. So there are 3 parts to the monomer of a nucleic acid. We'll start here. So number 1, you'll see it's a ring with an O in it. What are the other molecules in the other corners? If it's not written, these are what? Each of these in the ring are a carbon. So we have carbons and an oxygen in a ring shape. What kind of molecules often have ring-shaped carbons that we've talked about already? Yes, organic, I don't disagree. Which kind of the organic molecules? Carbohydrates, specifically a sugar. So a nucleotide has 3 parts. One part of that is a sugar. These sugars are pentose sugars. Ose tells us it's a sugar. Pent tells us what? It has 5 what? 5 carbons. So it's a 5-carbon sugar. You'll notice that the 5 carbons are not in the pentagon-shaped ring. We have 1, 2, 3, 4, and then the 5th carbon is outside the ring, but it's still considered, it's part of the sugar, so it's still a 5-carbon sugar. So every nucleotide has a 5-carbon pentose sugar. Second part. Again, has a ring structure, and they didn't put anything in there to help you figure out what it is, but this ring at least has 1 nitrogen in it. These are the nitrogenous bases. I did spell that right. So it's a base, so that tells you that it's got a little bit of a charge to it. A positive or a negative charge. Other one? Yeah, positive. So it's got a little bit of a positive charge on it, and it has a nitrogen in it. That's what makes it a nitrogenous base. There are 4 of those. So these are the 4 nitrogenous bases in DNA. There's actually 5, sorry. Okay, so you'll notice these 2 down here, and I wish I had a great way to help you remember this. I have not come up with one yet. Again, you can see we've got lots of nitrogens in here. These 2 have a double ring structure. Those 2 rings, these are called the purines. So these are the purine nitrogenous bases are A and G, adenine and guanine. Again, they have a double ring structure with nitrogens embedded in them. Lots of double bonds. The other 3 that you see Up here, you'll notice only have one ring. Again, lots of nitrogen still. These are pyrimidines. That's an N. The pyrimidine bases. Notice there's 3 of them. So there's cytosine and thymine and uracil. So cytosine is in both DNA and RNA, but thymine is only in DNA and uracil is only in RNA. So those two will swap out if you're talking about DNA versus RNA. So those are the nitrogenous bases. That's the second part. And then the third part over here, this group, you should recognize as what? What functional group is that of your 7? Somebody said it. Yeah, phosphate. So every nucleotide has to have a phosphate group, and that can vary in number from 1 to 3. This one only has 1. You can have anywhere from 1 to 3 phosphate groups attached. And you'll notice that has a little bit of a negative charge on it, because those oxygens have negative charges on them. So some people think of it this way, if this helps you, great. That every nucleotide has a house, because this looks kind of like a house. Because it's like got the roof, it's a house. And then it has a driveway. I don't know why this is called a driveway, except that we need a driveway. And then a pool. Because the phosphate group, P, pool. Has to have all 3 parts. So this is one monomer here. You can see this is showing you how that fits in here. So this is showing you what we call the sugar phosphate backbone. So when they bond together, you have the phosphate group bonding to the sugar. So the phosphate bonds to a sugar. So you have this alternating pattern down the side of the nucleic acid, down the backbone, sugar, phosphate, sugar, phosphate, sugar, phosphate. And then the bases are sticking out into the center. So you have here, this would be a purine base, and then these are all pyrimidine bases because they only have the 1 ring. So we have a sugar phosphate backbone with the bases sticking out. That's our polymerization. So how do we stick them together? What kind of reaction sticks them together? Dehydration synthesis. And the kind of bonds that are being formed between the sugars and the phosphates are phosphodiester bonds. So that's the kind of bond that you should associate with a nucleic acid. So the sugar and the phosphate are bonded together with a dehydration synthesis reaction that creates a phosphodiester bond. So we're going to talk about the 2, oh, whoops. Sorry, there's the sugars. There's 2 different sugars. So deoxyribose is the sugar in DNA and ribose is the sugar in RNA. Again, both of them are pentoses. And we're going to talk about the 2 different ones here. So DNA stands for deoxyribonucleic acid. That's your DNA, deoxyribonucleic acid. So the deoxyribo is telling you the sugar in this case, the sugar that's found in DNA is deoxyribose. And that's one difference that we'll talk about between DNA and RNA. You'll also see the structure of DNA is what we call a double helix. So it's got 2 sugar phosphate backbones. And we'll talk about this again when we talk about DNA replication. It's a pretty flexible structure. It's got some flexibility to it, but we tend to redraw, we show it like this, but it can be like this, and it can be all the way like this. But it's a double helix structure, so I think it like a twisted ladder. And when we talk a little bit more about this, it's chapter 9 or 10. We'll talk about how we discover the structure of DNA and how we know that what we know about the structure of DNA, some of the history behind how we know this. We talk about the strands being anti-parallel. So they run in opposite directions, which may not be totally and completely intrinsically obvious, I guess. But because of the way the bonds are oriented in the sugar phosphate backbone, you end up with what we call the 3' carbon at one end bonding with the 5'. So this strand runs 3' to 5', and this strand runs 5' to 3'. When we talk about the 3' carbon, these are the ones that have the OH group. The 5' carbon have the phosphate group. So one strand runs 3' to 5'. Again, that 3' and 5' is just referring to the carbon numbers, while the other one runs 5' to 3'. So they're anti-parallel because they're not going in the same direction. They're not both going 3' to 5'. And you're going to see specific what we call complementary base pairing. Complementary. So the sugar phosphate backbone with the nitrogen bases sticking out, the nitrogen bases are always going to pair up in the same way. So each rung of the ladder has 2 bases. So C always pairs with G and A always pairs with T. So those of you who are from this area might remember it as, actually, if you write it this way, it helps better. At Gates Chili, that's how I sometimes remember it. Gates Chili goes together. That's a school district in the area. A goes with T, it's a word that helps you at, and the other one is not a word. But A always pairs with T, C always pairs with G. And guess what kind of bonds hold the base pairs together? Hydrogen bonds, yes. So these are hydrogen bonds holding them together. You always have one purine base and one pyrimidine base going together. So one of the 2 rings and one of the 1 ring bases. That's the way it fits together in the strand to make those rungs together. And DNA is found where in the cell? I know we haven't talked about the cell yet. That's next chapter. This is all in the nucleus. It never leaves the nucleus if the cell has a nucleus. And we already talked about the fact that it's self-replicating. The other nucleic acid that's really important that we're not as familiar with is RNA. Again, the name is going to tell us something about it. RNA. So ribonucleic acid is a little different than DNA. In this case, our pentose sugar is ribose rather than deoxyribose. And this is going to look different than DNA. Sorry, this takes a second. It doesn't always come easily. It won't come apart all the way. It is a single helix. So we're imagining we just see one side. Okay, so there's just one side of the ladder with one nitrogenous base sticking off most of the time. So it's single stranded. The 4 bases that we have here are A, U, G, and C. So there's no thymine. There's uracil instead. So that's the one that is different. I said most of the time it's single stranded. It's not always single stranded. Sometimes the RNA, different types of RNA molecules will pair up again with hydrogen bonds to complete their job, which we're not going to talk about in a whole ton of detail right now. Just knowing that there are different kinds of RNA. So there's RNA that's called mRNA, M meaning messenger. So messenger RNA is found in the nucleus. It's made in the nucleus and then leaves the nucleus, so it's carrying the message. Then there's tRNA and rRNA, which are involved in making the proteins. So those are just different forms of RNA, but all of them are ribose sugar and these 4 bases. And we'll talk again a lot about these when we're talking about the details of the central dogma of biology, that DNA makes RNA and RNA makes proteins. Okay. Questions about any of that? Okay, that's the end of chapter 5. So we're going to just talk a little bit about the test, some test taking ideas, and then we're going to do some practice questions for the test for Friday. So the test is written now, so I can tell you a little bit more about it. I didn't have it today, so I'm Monday. There are multiple choice questions. I think there are 18 multiple choice questions. There's some matching questions, 10 matching questions, and then short answer and diagram. So there's not any like long essays that you have to write. I think one of the short answers you might have to write a couple of sentences for, but most of them are fill in the blanks kind of things, looking at a picture and trying to interpret information that you have. Questions about that at all? There are 18 multiple choice, 10 matching, and I think there are 12 short answer ones. Again, most of them though are not like 10, 15. 2 of them actually matter. But they're not. So just some general test taking strategy kind of things. I know you've all taken a test before. Maybe not for a while though. You had tests already this semester? No, so it's not be the first test. So just some reminders. When you're taking a test, read the questions carefully. You should be writing on the test. Okay, when you're taking a multiple choice test, you should be crossing things out, highlighting things, circling things. What is the key word in this question? This is a choice I know isn't right, so that if you have to skip it, you can come back to it and be like, oh yeah, I already thought through this and I remember what I thought through. Okay, you're gonna have to work efficiently. Hoping you're not gonna be like super stressed, but you're gonna have to work efficiently. So make sure you're writing on the test as you're taking it. Give yourself some notes, cross out things that you know are not right, circle words or highlight words that you think are important. If you wanna come in with highlighters, I'm fine with that. Okay, if that helps you, that's totally fine. To be writing on the test with highlighters, and if that helps you think through things, that's fine. Skip questions and come back to them. Do not get stuck on a multiple choice question. Okay, don't spend 10 minutes on one multiple choice question. If you're feeling like you don't know what it is, just skip it and come back to it. If you took a couple notes on it, crossed a couple things out, and you still don't know, skip it and come back to it. And then start with sections you feel more comfortable with. Some people love multiple choice and they just want to do multiple choice and they feel better about life when they've gotten multiple choice out of the way. Some people go right to the short answer because they're like, yeah, what's gonna be on the short answer and I just want to get those out of the way. You don't have to do it in order, so think about what works for you, what works best for you, what kinds of questions you feel more comfortable with. Sometimes use your test as a resource also. Sometimes there are answers on the test, one part of the test for the other part of the test. So if you're struggling with the question, go back and look at the multiple choice questions. Maybe some of the information that you need will be there that will be helpful for you. That's a good way to use your resources. So make sure that you're thinking about that as you go. So questions about any of that? We're going to practice some test questions together, and we'll go through some of these test taking things too. So the question will be on the screen, write down your answer, and then we'll talk about it. You can talk to the people next to you because this is not a real test. I tried to give you like hints about what category where we were thinking about this. So we're thinking about atomic structure. And I'm gonna talk to you about my thought process when I'm looking at a question too. So the first part of the question says a blank has a charge but negligible mass. So I know I'm talking about something that has a charge. What are my two choices? It's a proton or an electron. Negligible mass means what? You can't even notice it. So which one is bigger? Protons are bigger, so if it has negligible mass, we're talking about an electron. Okay, this one has mass but no charge. Keyword is no charge. What is it? It's a neutron. It doesn't have a charge. Okay. So this might be an example of something that might be more like a short answer diagram question. Again, it's not long answers, but there's a diagram here for you to look at and think about. So take a minute to think about this, talk to the people around you.
We do that.
If you need a periodic table, there will be there. Please do not memorize it. But the information that you would need, it would be on a periodic table.
Did I give it for you too for this question? No, but. Okay, what atoms is this? Carbon, we know that. Why? How many valence electrons does it have? It's got 4 valence electrons and it's our favorite atom anyway. So it's always the answer. Not always the answer, but. Okay, so it's got 4 valence electrons. It's carbon. How many bonds can it make? 4, because how many electrons does it want in its outer shell? It wants 8, so it's going to make 4 bonds. What kind of bonds does it make? It could be single, double. Oh, my internet. Single, double, or triple. What kind of bond? Covalent bonds. It's going to share the electrons. And are they polar or non-polar for carbon? It's kind of a trick question. It can be either, because it depends on what it's bonding with. Okay, we talk a lot about carbon-hydrogen bonds. Those are non-polar bonds. But carbon can make polar bonds depending on what it is bonding with. I think it talks about definitions of polar and non-polar. I don't know that it specifically asks about if it's polar or non-polar for a specific bond. I'm not guaranteeing that, but I think that's what I remember. But you should know that carbon and hydrogen are not polar. That's not a polar bond. That makes molecules that have a lot of, a lot of carbon and hydrogen. A hydrocarbon is non-polar. So what is it going to be, hydrophobic or hydrophilic? Hydrophobic. If it's non-polar, non-polar leads to being hydrophobic. So a non-polar bond is going to be hydrophobic. What's an example of a molecule that has lots of carbon-hydrogen bonds that is hydrophobic? Which of our 4 types of molecules has lots of carbon-hydrogen bonds and is hydrophobic? Lipids. Lots of energy in those bonds, but hydrophobic. Maya, did you have a question?
We use electronegativity trends, like how like fluorine is like really electronegative compared to like fusion bonds, or do we just
I think you should probably know that like bigger molecules tend to be more electronegative, but in terms of like it's increasing as you go right and down on the periodic table, we didn't talk about that even. So just in general, bigger is more electronegative is enough information, I think. Okay, chapter 3, we talked about water. What are the 4 emergent properties of water? Take a minute to think about it. Jot them down.
If I remember. Okay, somebody give me one. What is one emergent property water? How about somebody really talk shit today? Okay. I've been tempted to write drains of possible six, but I haven't done it.
Okay, one is cohesion, good. Cohesion, meaning what? It's gonna stick to itself, right? What kind of bonds make water stick to itself? Hydrogen bonds. Sometimes water doesn't stick to itself, sometimes it sticks to other things. What do we call that? Adhesion. Also caused by what? What kind of bond? Hydrogen bonds. Okay, because that partial negative and partial positive charges on the water molecule can stick to other partial positive and partial negative charges in other kinds of molecules. That also leads to the high surface, whoa, that's not tension. Tension of water, but the main one when we talk about the four emergent properties, or really cohesion is the one that we name the most, but they're all related to each other. So that's one of them. How about another one and another person? So moderation of temperature, because water has what kind of bonds? Hydrogen bonds. It has a really high specific heat, so it has the ability to absorb a lot of heat, a lot of energy before it changes temperature. So because of that, that helps us regulate our body temperature, that helps aquatic ecosystems regulate their body temperature, and that also helps to regulate our climate. That's the same idea. As long as you get cohesion, you're fine. So you know that.
Water is less dense as a solid than it.
Yeah, so density. This is unusual. It has less density when it's a solid rather than a liquid. Why? What kind of bond makes this happen? Hydrogen bonds. Always the answer for water, okay? As the water molecules are moving around in a liquid, they have hydrogen bonds that are breaking and reforming, but as they start to slow down, as the temperature decreases, they start to slow down, and those hydrogen bonds get more stable, and that actually pushes the water molecules apart a little bit more, so water is 10% less dense when it's frozen than it is when it's solid, when it's liquid, sorry. It's 10% less dense when it's a solid than when it's a liquid, and that allows it to float. We talked about that's important because that keeps aquatic ecosystems functioning. So that's 3. What is the 4th one? I'm hoping somebody remembers because my brain is not working right now. Thank you. We call it the universal solvent. Does it dissolve everything? No, but because it's polar, it's a polar molecule, it can dissolve a lot of things. And that's important because that allows us to cause a lot of reactions, to move materials around, that kind of thing. And its polarity is the polar covalent bond, but also makes hydrogen bonds too. You have related to hydrogen bonds. Okay, so those are the 4 emergent properties of water. That's what makes water special. Oh, here's an easy one. What type of bond gives rise to the four emergent properties of water? Hydrogen bonding, yes. Okay, and the key to this should be you're seeing bond and water. So there's only two kinds of bonds that you should think about when you're thinking about water. One of them is hydrogen bonds, and the other one is polar covalent. Because it's got that big oxygen and the two little hydrogens. The bond is polar. We get a partial positive around both of the hydrogens and a partial negative around the oxygen. Ionic bonds, we're not thinking about when we think about water, except if we're dissolving them. It's not nonpolar, so we're not thinking about that. And disulfide bridges are very specific. We're thinking about that when we're thinking about the tertiary structure of a protein, not water. Okay. So, starch and cellulose. So let's think about our answer questions working from the bottom up. Starch and cellulose are structural components of the plant cell membrane. What do you think of when you think of membranes? What molecule do we think of? It looks like this. So membrane should make you think of phospholipids. So our starch and cellulose phospholipids. So we're not going to care about that. Cis and trans isomers have to do with what kind of bond? Carbon double bonded. So is that possible? Maybe. But we're thinking about OS, which we know is a what? We know this is a sugar. So when we think of double bonds, we're probably not thinking too much about sugars in terms of this. Although we could, I guess. Are both used for energy storage in plants and animals? Okay, when we think of cellulose, we think of what? Plants or animals? Plants. So do we think about animals with cellulose at all? No, so we can already eliminate that. So maybe we're thinking about this one, or we're thinking about this one. Polymers of glucose. Glucose is a what? Sugar, it's a, what kind of saccharide? Monosaccharide, so that would make sense, right? We have a monosaccharide, we can make polymers out of it, and we would probably know, hopefully, that these are polymers of glucose. So maybe a longer thought process than you need to. You don't need to overthink them all. If you are confident on it, don't sit there and try to think through all the answers. If you know it, you know it. Okay, this one I put up here, this will not be on the test. This is harder than a test question, but I wanted to show you how to think about this question in a way that would make it less intimidating. Okay, it says use the figure to answer the question. Which molecule can be a result of a mercapto ethanol reduction of a disulfide bridge? Did we talk about the mercapto ethanol reduction of a disulfide bridge? No. Okay, so this could induce panic in your life if you saw this question. So what I want you to think about is what is the key word in this question that should not make you panic? What is something that looks familiar to you? Disulfide bridge. Okay, disulfide tells you two sulfhydryl groups. So which one of these has a sulfhydryl group in it? B. Okay, so even if you don't know anything about the question, you don't know what the mercapto reduction of a disulfide bridge is. Maybe you do, but I don't want to underestimate you. But even if you have no idea, if you can pick out that keyword of disulfide bridge, and you can say disulfide, oh, I remember that has something to do with sulfhydryls. This doesn't have any sulfur. This doesn't have any sulfur. This doesn't have any sulfur. This one has sulfur in it, so it's got to be that one. And that is the right answer. Mercapto is just, ethanol is just a chemical that can break a disulfide bridge. So that would leave one sulfhydryl group. But again, you don't have to know that to be able to answer that question. Which of the following descriptions best fits the class of molecules known as nucleotides, which we just talked about? This should be real fresh in your mind. How many components are we looking for in a nucleotide? 3. So we can already automatically eliminate this one, and that one. You don't even have to know what they are. You just have to know there's 3 things in a nucleotide. And what are they? They're a nitrogenous base, a phosphate group, and a sugar. It's the house, the driveway, and the pool. Okay? So you don't have to know the answer even, you just have to know what you're looking for. Which of the following best summarizes the relationship between dehydration reactions and hydrolysis? Okay, so let's think about our choices here. Hydrolysis reactions create polymers and dehydration reactions create monomers. Okay, we know about monomers and polymers. Some of that makes sense. Why does this answer not make sense? What does hydrolysis mean? Hydro means water, and lysis means breaking down. So when you lyse something, you're breaking it. So a hydrolysis reaction is going to use water to break something. Is it going to create something? It's going to break it down. So dehydration, what are we taking away when we dehydrate something? We're removing water to create monomers? No. Okay, so we're not going to be okay with that answer. Dehydration reactions convert sugars to lipids, and hydrolysis reactions link fatty acid chains together. So again, hydrolysis means breaking apart, so we're not going to be putting things together. Did we talk about converting sugars to lipids at all? No, we're not there. Dehydration reactions and hydrolysis reactions both assemble polymers from monomers. Do they both make polymers? They're opposites of each other, so they can't be doing the same thing. So that means it has to be this. Dehydration, we know, is dehydration synthesis to make polymers. Hydrolysis breaks polymers. So it has to be that one. Hey, we've got 2 minutes. Where are you going? Where would you find a glycosidic linkage? Which kind of molecule is going to give you a glycosidic linkage? What holds 2 water molecules together? Hydrogen bond. Okay, why do we think hydrogen bonds? 2 sulfur groups together. The disulfide bridge. 2 glucose molecules. Glycosidic. Glycosidic looks kind of like glucose. That's going to be our answer. What kind of bond holds 2 amino acids together? That's a peptide bond. Okay. I have one more slide that I wanted you to see, but it's not going. There we go. Okay, so when you're studying between now and Friday, things to think about. On the lecture syllabus, it tells you which sections are we covered. There are parts of chapters we didn't cover that you don't need to study, so make sure you're looking at the syllabus. Look over your notes. The PowerPoints are on Google. You can print them out or link them on your tablet, diagrams and things like that, right all over them. Chapter outlines on Moodle, you can use those to fill those in. That might help you study flashcards. Tell somebody how and when you're going to study because you have a test on Friday. A lot of you did not sign this. Lots of you did not get it.
I will be taking my test in the Learning Center.
Okay. I need them to tell me that.
Okay, I already did that.
Okay,
So
like, did you set up a time?
yeah,
Okay, so they should be emailing me, so they
Today.
will have a copy of the test.
Yeah.
Perfect.
just letting you know before.
No, I knew that there were a couple people that did, but I hadn't heard anything from the Learning Center. So
Yeah.
I'll make sure that I hear from them.
Yeah.
Not a problem at all.
They can be very late. On these filings sometimes.
I know.
They emailed me though, so
Okay, good, good.
they should email you.
Okay. I did check this morning thinking I could hear something, but I haven't.
It's in your folder in the left.
So I just didn't know like the shit, what if we were trying to get.
You don't need it though.
Even the word that tool, yes.
Milestones, you can look at it.
What point do I have it? It's gonna be from. Right.
Can't tell from that. That was more than you
Just
did.
look at it.
Yes. Eventually, if you work with it enough, like you start to memorize them, but that it's not something
Yeah.
you that you memorize. Really, you should know just.
Okay, so it's like, how do I?
No.
And then I'll, I was on this, so I'm used to it.
Was there one that didn't see like this?
Yeah, so I looked at my.
Yes.
But one of the questions.
If you're not. I got a look or and it structure. Yeah. Gotta figure out who that is first.
There, Woodford.
Arm got it. I got a name.
I do try to. Once I have not completed it, it should show up. That's possible. I mean, but that would be like your short answer, one maybe, or.
Okay, I want you to stop recording.